Okay, I think we can start now. Am I audible? Okay, I hope my slides are visible. So let's start the session. Welcome to Host Pathogen Interactions by NPTEL. This is week 7 live session. The course instructor is Professor Himanshu Kumar from ICER and I am Sumati Raviraj, TA from University of Hyderabad. If you guys have any doubts or any queries, you can mail me regarding the same. So for week 7, sorry, yeah. So for week 7, uh, the syllabus was complement system. So it is, yeah. Can you please mute your mic? So first there were these topics that was covered, that is your innate immunity, complement introduction, complement system, all the three pathways, that is your classical pathway, alternative pathway, lectin pathway, and how complement system regulation occurs and how it leads to certain diseases. And then there was introduction to adaptive immunity. So coming to the weekly assignment, so we'll just go through the basic pathways once before going into the particular topics regarding each question. So first we will see about alternative complement pathway. So alternative complement pathway is a defense mechanism that helps the body identify and destroy microorganisms. It's part of the innate immunity and it is a natural defense mechanism against infections. So the basic steps of alternative pathway is C3b molecules that bind to the activating surface, and then factor B binds to the surface bound to C3b, and B is cleaved, factor B is cleaved within C3b complex. And how it works is when it is activated by virus, bacteria, fungi, parasites, and other foreign materials. It can also be activated by spontaneous hydrolysis of complement C3. Then opsonization of microorganisms is done by C3b, and its effectiveness relies on its ability to distinguish self from non-self. So C3b is one of the major, C3, C3b is the major component of your alternative pathway. And then first is the binding of the complement proteins to microbial cell or to any antibody, and then formation of C3 convertase enzyme. Then cleavage of C3 occurs, that is hydrolysis, and then formation of C5 convertase. So you see C3 again has C3b, and then you also have factor B, and then finally you get C5b and C5a. So then we have the classical pathway, in which there are some additional steps, and from C3 again it is similar to the alternative pathway. So the classical complement pathway is a part of the immune system that is activated when antibodies bind to antigens. The pathway is one of the three ways that can activate the complement system, that is your classical, alternative, and lectin. So in classical complement pathway, first, the C1q protein binds to the Fc region of the antigen antibody complex. So here we can see we have the antigen antibody complex, and the C1q part of the C1 binds to this Fc region of this complex. then C1R is activated, and then this cleaves C1S. Then C1S binds to C4, which is enzymatically cleaved into C4A and C4B. So from C1, you get C4A. This is again cleaved to get C4B and A. Then further C4B binds to C2, which is cleaved into C2A and C2B. Then C4B and C2A form a complex, which is the C3 convertase. And now this complex cleaves the C3 into C3A and C3B. After this, C3B binds to C4B 2A complex to form C5 convertase complex. So now the C3A will bind to this complex and form C5 convertase complex. Uh, then C5 convertase cleaves C5 into C5A and C5B. C5B then initiates the formation of membrane attack complex, that is the MAC formation. Then the classical complement pathway attracts phagocytes and initiates the phagocytosis through them at the site of infection and tags the target cell for elimination. So this is the classical pathway. Next is the lectin pathway. So lectin pathway is part of the body's defense mechanisms that activates the complement system, similar to the other complement pathways. It's the main binding factor here is the mannose. So mannose binding lectin, that is the MBL. Mannose binding lectin, and we also have collectin 11 and then ficolins. So these bind to the mannose or the carbohydrates residues that are expressed on the pathogens. These proteins then form a complex with the MBL associated serine proteases. These are called as MASTs, MBL associated serine proteases. Then again we have C4 and C2 that are cleaved. Again similarly, similar to the classical pathway, we have the C4B2A complex formation, which is a C3 convertase. Then C3 is cleaved, forming the membrane attack complex. So here we have again C3B, C3A, and then again we have C5 convertase complex, and then C5 convertase again gives rise to C5B and C5A. And finally, we have MAC complex formation. It opsonizes and tags foreign cells for cytosis, induces inflammatory reactions, lyses target proteins, and cleaves protease activated receptors. So this is the gist of all the three pathways: alternative, classical, and lectin. So C3 is major, common in all of them, and downstream again all the processes are similar. Activation of each pathway differs based on uh, the stimulus. That is, for lectin pathway we have carbohydrates or mannose that is present on the pathogen. For classical pathway we have the antigen antibody complex, and then here in case of alternative pathway, C3b molecules bind to the activating substance or any foreign material directly. So coming to specific topics related to the questions, first we have about C3 convertase. C3 convertase is a key enzyme complex in the complement system, and it is responsible for cleaving C3 into C3A, which is an anaphylatoxin, and C3B. C3B participates in opsonization, immune complex clearance, and complement activation. It plays a central role in all the three complement pathways, that is the classical, alternative, and lectin, but differs in the composition depending on each pathway. So in the classical pathway, C3 convertase is C4B and 2A. The formation is activated by antigen antibody complexes which bind to C1Q, leading to cleavage of C4 and C2. So C4b binds to pathogen or immune complex. C2a is a serine protease that is responsible for C3 cleavage. So C2a, C4b fragments, then we get the C3 convertase, then C3 hydrolysis, then C3b, C3a fragments, then we have C3b which cleaves C5 into C5a and C5b. So the C3, which is cleaved into C3A, C3B, facilitates the C5 convertase formation, that is a complex of C4B, 2A and 3B. So this continues the complement cascade downstream. Then coming to the alternative pathway, C3 convertase. Here C3 convertase has C3B and factor B. So formation of this, the pathway is activated spontaneously by low-level hydrolysis of C3. This is called as C3 takeover, which allows C3b to bind to surfaces lacking the regulatory proteins. So now C3b binds to the pathogen surface, then factor B binds to C3b and is cleaved by factor D, generating C3b factor B complex. Then we also have factor P, which is properdin. This can stabilize the complex. So the major function is cleavage of C3 into C3b, leading to amplification of the pathway and formation of C5 convertase. So here in alternative pathway, C5 convertase complex has C3b, factor BB and again 3B. So now we can see here how different pathways uh, lead to C3 hydrolysis. For classical, you have C1, C4, C2, and for lectin, you have your MASP, speculins, MBL complexes, and for alternative, you have direct uh, spontaneous hydrolysis of C3, and then from C3, and then you have factor B. And finally, everything leads to C3 convertase complex formation and C3 enzyme cleavage occurs, forming C3A and C3B. Then in lectin pathway C3 convertase, we have C4B2A, similar to the classical, and it is activated by mannose binding lectin. So activation differs here. For classical, it is antigen antibody complex, and for lectin pathway, it is the mannose residues on the pathogens. So it is activated by mannose binding lectins or ficolins, binding to the pathogen associated carbohydrate structures. This activates MBL associated serine proteases which cleave C4 and C2. So the composition of the C3 convertases C4B2A. C4B attaches to microbial surface and C2A is enzymatic subunit that cleaves C3 enzyme, a C3 component of the complement. Then cleavage of C3 happens, generating C3b to enhance the opsonization and formation of C5 convertase complex, which consists of C4b, 2a and 3b. And then finally C5 convertase cleaves C5 component into C5a and C5b. So then MAC complex formation occurs and leads to phagocytosis or lysis by recruiting phagocytic cells. So each C3 convertase leads to C5 convertase formation, triggering C5 cleavage, membrane attack complex formation and subsequent pathogen lysis. So in each case, we have C3 convertase which activates C3 and then subsequently C5 convertase formation and then triggering cleavage of C5 and then formation of membrane attack complex and then pathogen lysis. So this is a gist of this, a table. So for classical, the activation and trigger is antigen antibody complex. For alternative, the activation trigger is spontaneous hydrolysis of C3. For lectin pathway, it is MBL or ficolin binding to the microbial carbohydrates. C3 convertase composition is same in case of classical and lectin, that is consisting of C4b and 2a, and in case of alternative it is consisting of C3b and factor B. Next is a significance of the C5 convertase in classical complement pathway. So C5 convertase is crucial enzyme in the classical complement pathway that facilitates the transition from C3 activation phase to the terminal complement cascade. So this leads to the membrane attack complex formation and cell lysis. So the classical pathway C5 convertase is composed of C4b, 2a and 3b. C4b and 2a, this is the C3 convertase complex, cleaves the C3 into C3a and C3b. C3b binds to C3 convertase, forming C5 convertase. So basically it cleaves C5 into C5a and C5b. So in this C5 convertase, we have C5a. This is a potent anaphylatoxin that promotes inflammation and recruits neutrophils, increasing the vascular permeability. Then C5b is the one that initiates the membrane attack complex formation by subsequently binding to C6, C7, C8 and multiple C9 molecules, forming a pore in the target cell membrane. So we have our C5 which splits into C5a, C5b. Then C5a is it helps in increasing the vascular permeability by binding to the receptor, and then neutrophil mobilization occurs, and some anaphylactic reactions leading to histamine release, smooth muscle contraction, tissue factor production, etc., is downstream of C5a action. And then C5b forms the membrane attack complex by binding to C7, C6, C8 and a multimer of C9. So this then forms a pore forming complex in the target cell, which then leads to cell lysis, apoptosis and cytokine production. Biological significance of this. This triggers the terminal complement cascade, that is the MAC complex, and creates pores in bacterial, viral infected cells or damaged host cells, leading to osmotic lysis and death. It enhances inflammation via C5a. So C5a is a powerful chemotactic factor attracting neutrophils and monocytes to the site of infection. It promotes the release of pro-inflammatory cytokines as well from the immune cells, and it also increases vascular permeability, helping the immune cells reach the site of infection. It also amplifies opsonization and phagocytosis. So the C3b fragment in the C5 convertase complex remains on the microbial surfaces, and this enhances the phagocytosis by immune cells with complement receptors CR1 and CR3. So basically triggers the C5 convertase, triggers the terminal complement cascade, enhances the inflammation, amplifies opsonization and phagocytosis. Okay. Hope I'm audible. If I'm not audible at any point of time, you can let me know. Yeah. So the next is IgM. So how IgM is the most potent stimulus, or it is the most effective antibody in initiating the classical complement pathway. So among all the antibody classes, that is A, G, D, E, M is the most effective because it forms a pentameric structure. So IgM exists as a pentamer in circulation, providing multiple Fc regions that can simultaneously bind to the C1q, the first component of the classical pathway. So here we can see the structures of different classes of antibodies. We have IgA, Ig3, then IgD, we have IgE and a pentamer of IgM. So this pentamer is connected with a J chain. So you can see this provides multiple FC regions so that it can bind to C1q of the classical pathway. So then high complement activation occurs, so the efficiency increases. So even a single IgM molecule bound to an antigen is sufficient to activate C1q, leading to C4 and C2 cleavage, forming C3 convertase. The pentameric IgM molecule bind to the antigens on the bacterial surface and adopt the staple form. So you can see here, this is in the circulation. This is the pentameric form in the circulation, which is also a planar form. Once it binds to the antigens, it takes a staple form. You can see how the structure of the pentamer has now changed. And IgG, a less effective complement activator. IgG is a subclass containing IgG1 and IgG3. This can also activate the classical pathway, but they are less efficient than IgM. So requirements for IgG to activate C1q, at least two IgG molecules are necessary to be close together on an antigen surface for binding with C1q. So you can see here a dimer that is two of the IgG is required, unlike here where we have a pentamer. So even though they exist in pentamer, IgM, even a single molecule bound to the antigen is enough to activate. So that is the reason why IgM is a better activator or a more potent activator. So then we have Ig3, which is greater than Ig1 and greater similarly than Ig2 in activating the complement system. So Ig4 does not activate the complement system. So we can see a gist of all the classes here of subclasses of IgG versus the IgM. So IgM is most effective, requires only one pentamer to bind to C1q. Ig3 is next to it, which is strong. IgG1 is moderate, IgG2 is weak, but Ig4 does not activate. So Ig3 requires a multiple IgG molecule. Ig1 is activates the complement system, but is less efficient than IgG3, and IgG2 also activates the complement system, but under specific conditions only. Next is the role of haptens. So haptens are a small molecule which is antigenic, but it is not immunogenic. So when we say it's not immunogenic, it cannot generate an immune response on its own. It requires a carrier molecule. So that is why it is a hapten. So this means that the hapten can be recognized by the immune system, but cannot trigger an immune response unless it is attached to a larger carrier protein. So majorly the characteristics of haptens are they are small molecules, and they cannot induce an immune response alone due to their small size. Many of the drugs, chemicals, toxins, all these are small molecules which have the antigenic property but cannot invoke an immune response on their own. So they become immunogenic when they conjugate with a carrier protein. So these carrier proteins can be present within the circulation like albumin, globulin, etc. So once they bind to this carrier protein, now they become a complete antigen to provoke an immune response. So now this immunogen or the hapten carrier complex will now bind to the antibodies or can stimulate the immune response. So immunological role of haptens, a hapten carrier complex formation is required. A hapten by itself does not activate neither B cell nor T cell, but because it lacks the size and the complexity required for the effective immune recognition. So only when the antigen is recognized properly by either the antibodies or the T cells or antigen presenting cells, it can invoke an immune response. So when a hapten binds to a carrier protein, it forms a hapten carrier conjugate, which then activates the B cells to produce antibodies and also triggers the T cell responses via antigen presenting cells. Role in drug allergies and hypersensitivity. Excuse me. So some drugs can also act as haptens and trigger allergic reactions. Example, penicillin binds to proteins in the body, forming a hapten carrier complex, which can trigger an immune response leading to penicillin allergy. So sometimes when antibiotics are prescribed, they also check whether particular person is allergic to certain antibodies or not, because it as few of the antigens or drugs, they also have antigenic property, and since they are smaller in size, they are haptens. But when within the body they bind to proteins like albumin or globulin, they can become immunogenic in nature. And also poison ivy, the plant we come across. So it has urushiol, which forms a hapten protein complex in the skin. This leads to delayed hypersensitivity reactions and contact dermatitis. So this can also, spread, and it can cause skin reactions, rashes, etc. Then the role of hapten in vaccine development. Some vaccines use hapten carrier conjugation to enhance the immune response. Example, conjugate vaccines like the Hib vaccine link a bacterial polysaccharide, which is a hapten, to a protein carrier to stimulate the T cell dependent immunity. And then it also has a role in diagnostics. So hapten based assays are used in ELISA, immunohistochemistry, drug testing, etc. Small molecules like hormones, drugs, or toxins can be detected using hapten antibody interactions. So these are few other examples of haptens: penicillin, urushiol from poison ivy, then DNP, dinitrophenol, bacterial polysaccharides, etc. So these can form complexes with carrier proteins like serum, albumin, globulin, skin proteins, BSA, which is bovine serum albumin, and then tetanus toxoids. All these are carrier proteins for different haptens. So penicillin allergy, contact dermatitis are because of these two. And then this DNP and BSA can be used in experimental immunological studies, and then bacterial polysaccharides and tetanus toxoids used for vaccines are Hib and pneumococcal vaccines. Next is hereditary angioedema. So this, and then it is primarily associated with C1 inhibitor, that is a complement C1 inhibitor. The disease, hereditary angioedema, that is HAE, is primarily associated with C1 inhibitor deficiency, leading to this condition. So what basically this is it is a genetic disorder that is characterized by recurrent episodes of severe swelling. So angioedema means swelling of the blood vessels. So angioedema in various parts of the body. So swelling commonly affects skin, gastrointestinal tract, and also the airways, which can be life threatening if it leads to obstructions. So if airway obstruction occurs due to these kind of swellings, then it can be suffocating and life threatening. Then the role of C1 inhibitor in immunology. So C1 inhibitor is a regulatory protein that controls the complement system, particularly inhibiting the C1 component of the complement. So C1 complex in the classical pathway. So it regulates also the other inflammatory pathways such as the bradykinin pathway, and also coagulation and fibrinolysis of by inhibiting the kallikrein. So bradykinin pathway by inhibition of kallikrein and factor 12, and then also in coagulation and fibrinolysis. Then deficiency or dysfunction of C1 inhibitor leads to excessive bradykinin production because it is involved in the regulation, and then causes increase in vascular permeability and tissue swelling, leading to several other uh, symptoms or several other complications because of the swelling, leading to obstructions. So, C1 inhibitor deficiency, recurrent isolated angioedema. Other than this, laryngeal edema occurs, recurrent abdominal pain, vomiting, etc., are part of C1 inhibitor deficiencies. So since this is a genetic disorder, it can also have, like, it can be passed down, and it has a positive family history will be there. And serum complement for C4 levels and C1 inhibitor functions are diagnosed. So if it is normal, repeat the analysis during acute attack. If it's still normal, then consider causes of angioedema other than type 1 and type 2. So there are again types of HAE. So it can be different because of ACE1, or it can be type 3. So in the initial cases itself, if the C4 level or C1 inhibitor function decrease, then we have to go through with the C1 inhibitor antigenically and see if it is normal or decreased. Again, if it is normal, it can be type 2, and if it is decreased, it is type 1. So this is basically how the different types of HAE are detected, because it can involve C4 levels, and basically the effect of C1 inhibitor on the complement pathway is checked to find out which type of HAE is also present. This is just a information, it is not required. So the clinical features of HAE are recurrent non-itchy swellings on the face and extremities, genitals, GI tract, airway, etc., severe abdominal pain due to swelling of the intestinal wall, no response to antihistamines or steroids, unlike allergic angioedema. So, angioedema can also occur because of other allergic conditions or reactions, but in those cases, antihistamines or steroid based drugs can help. But if there is no response with this, it can be hereditary or it can be HAE. So laryngeal edema is also one of the features. It can be fatal due to airway obstruction. So like in the previous, I explained the diagnosis. Low C1 levels is type 1, type 2 based on the activity, and low C4 levels during the attack, you can further detect it into AC1 or type 3 HAE. So genetic testing for SERPING1 mutations can be done if needed. Treatment involves acute attack, C1 inhibitor replacement therapy is given, and bradykinin receptor antagonists are given because that is also dysregulated now. Then kallikrein inhibitor, then supportive care for airway protection, and for long-term prophylaxis, C1 inhibitor concentrate for few frequent attacks, and androgens to increase the levels of C1 inhibitor, antifibrinolytics, and monoclonal antibodies, etc., to block the kallikrein and regulate it. Just So next is IgM. Why is IgM considered more potent than IgG? So previously in the previous question we discussed about it. So IgM is more potent than IgG in activating the classical complement pathway because of its structural and functional advantage in binding to C1q component of the classical pathway. So pentameric structure, it circulates as a pentamer. 5 antibody units are joined by a J chain, giving 10 antigen binding sites. So basically antigen binding sites are more. It can effectively bind and stimulate the C1q. So this structure provides multiple Fc regions that can simultaneously bind to C1q, the first component of the classical complement pathway. Efficiency in C1q binding. C1q requires at least two Fc regions to be bound simultaneously to activate. A single IgM molecule is sufficient to bind activate C1q, whereas at least two IgG molecules are needed because C1 IgM is already in the circulation in its pentameric form. Then functional implications, early immune defense. IgM is the first antibody produced during infection, primary immune response, and its ability to rapidly activate complement enhances opsonization, it enhances pathogen clearance, and it also enhances inflammation. So we can see here again IgG, you have it goes and binds, and after binding then it requires a minimum of 2 to activate the C1q of the complement. But since IgM is already in a pentameric form, a single IgM molecule is sufficient for the C1q component to come and bind to it. So IgM and IgG both activated. IgM is more potent, the staple form and the planar form helps it, and C1q binds to one bound IgM molecule enough to activate the system. C1q requires at least two IgG bound to the antigen to uh, trigger the complement cascade. IgG complement, like we discussed before, Ig3 is greater in activation than Ig1, and then we have Ig2. Ig4 does not activate. So why IgM is more potent is because of the pentameric structure, multiple Fc regions available, single molecule is enough, and then rapid activation, and immediate pathogen clearance. Next is the role of C3 in alternative pathway. So the alternative pathway is the part of innate immune system. It does not require antibodies. Instead, it relies on spontaneous hydrolysis of C3 to initiate the complement activation. So the major steps involved in the spontaneous hydrolysis uh, being the key here, the tick over mechanism it is also called. So the C3 undergoes slow spontaneous hydrolysis in plasma, forming hydrolyzed C3, and this allows binding of factor B for the alternative pathway. This is then cleaved by factor D, forming C3 convertase, giving rise to C3b BB, and then C3b convertase formation leads to C3 cleavage into C3A and C3b. C3b then binds to microbial surface similarly, and then amplifies the complement response via a positive feedback loop. So this amplification is very important, and formation of C5 convertase occurs with C3b, BB, and C3b again. So additional C3b molecule binds to the C3 convertase, forming C5 convertase. This now cleaves the C5 into C5a and C5b. C5b then initiates the membrane attack complex. So we have, you can see here the spontaneous hydrolysis of C3, then C3 convertase formation via factor B, then C3b, 3, C3b of the factor B, then we have formation of C3 convertase which cleaves C3 again, more C3, then C3b, BB, and C3b complex is formed which is a C5 convertase, and then it cleaves C5 into A and B. B triggers the MAC complex formation again by binding to several other complement components, and C5A is a inflammatory mediator. So the function of C3 in alternative pathway is to amplify the complement activation via positive feedback loop. You can see the positive feedback loop, and then opsonization C3b binds to pathogens, enhancing phagocytosis, inflammation, uh, because C3A is an anaphylatoxin. It recruits immune cells, and MAC formation occurs by C5b, and C5A also is a inflammatory mediator. So the role in lectin pathway is it triggers the recognition of pathogen associated carbohydrate patterns, that is the mannose binding lectins or ficolins. So MBL or ficolins bind to the microbial surface. So it binds basically to the mannose residues on bacterial, fungi and virus, and then we have the MASPs, the serine proteases. Then C4, C2 activation, similar to the classical pathway, C4A, C4B. Then C4B binds to pathogen surfaces here and recruits C2 and cleaves C2 into C2A, C2B. This forms the C3 convertase here. So C3 convertase over here is the C4B and 2A complex. Now this will cleave the C3, forming C3A and C3B, and then we get the C5 convertase C4B, 2A and 3B. Then we have C5 cleavage. So again similarly C5B we have MAC formation. So this also amplifies complement activation through classical C3 convertase and opsonization, inflammation and MAC formation. So opsonization, C3b coats the pathogens, enhancing the phagocytosis. So inflammation, C3a recruits the immune cells at the site of infection, and MAC formation via C5b. Then so role of C3, we can see uh, in both alternative and lectin pathway. Basically, it is amplification, opsonization, inflammation, and MAC formation. Then next we have professional antigen presenting cells. So professional antigen presenting cells, APCs they are called, are immune cells specialized in the processing and presenting the antigens to activate the T cells. So they express major histocompatibility complexes, that is MHC class molecules, and they have MHC class 2, allowing them to present the antigens to CD4 plus helper cells, CD4 plus helper T cells. So the major types of professional APCs are the dendritic cells and macrophages and B cells. So these are the professional antigen presenting cells. Their primary function is antigen presentation. So for dendritic cells, it is the most potent APC and it initiates T cell activation. It has high class MHC2 expression and strong cytokine production and activates the naive T cells. Macrophages engulfs the pathogens and presents antigens to the effector T cells activated by IFN gamma, involved in inflammation and phagocytosis, because macrophages are also phagocytic cells. B cells are also antigen presenting cells, so presents the antigens to the helper T cells for antibody production. And other than that, BCR mediated endocytosis can also occur and helps in the humoral immunity. So the role of professional antigen presenting cells in immunity is antigen uptake, antigen processing, co-stimulatory signals and cytokine secretion. So antigen uptake, after the APCs engulf the pathogens via phagocytosis, receptor mediated endocytosis or macropinocytosis occur. So this leads to uptake of the antigen. Then antigen processing. Once internalized, the antigens are then broken down into peptides and are loaded onto the MHC class 2 molecules. So then the T cell activation occurs because the naive CD4 T cells now recognize the antigen MHC2 complexes via T cell receptors. Then co-stimulatory signals are required for full activation. Example, CD80, CD86 binding to CD28 on T cells, etc. lead to co-stimulatory for additional activation. Then cytokine secretion. Antigen presenting cells release cytokines, many pro-inflammatory cytokines like interleukins, TNF alpha, etc., to shape the T cell response. So APCs are majorly dendritic cells, macrophages, B cells. They activate naive T cells, killing the pathogen by presenting the antigens. They present the antigen for the production of antibodies. Then the dendritic is the strongest APC. It migrates to the lymph nodes. Macrophages use phagocytosis and then release of cytokinesis. B cells have BCR response to capture the specific antigens. Then these cells are called professional antigen presenting cells because they express the MHC class 2 molecules and they provide co-stimulatory signals. They are efficient in processing and presenting the antigens to drive the adaptive immunity. So other cells other than the professional APCs also have the ability to present the antigens. So cells like fibrous, endothelial cells, eosinophils, mast cells, basophils, etc., can also present antigens, but they use MHC class 1 and they lack co-stimulatory molecules, making them non-professional APCs. So professional APCs are dendritic macrophages and B cells, critical for adaptive immunity, ensuring effective T cell activation, pathogen clearance, and immune memory formation. So you can see here professional APCs, we have the DCs, we have the macrophages, we have the B cells. So DCs and macrophages are phagocytic in nature. They express receptors for apoptosis. They recognize PAMs and DAMs. Then localize to specific tissues, localize to T zone of the lymph nodes, and following they have the activation, then constitutively express high levels of class 2 MHC and antigen presenting machinery, express co-stimulatory molecules after activation. Then for B cells, internalize antigens via BCR response, then constitutively express class 2 and antigen processing machinery. They also have co-stimulatory molecules which help in activation. Then atypical APCs, for example, mast, basophil, eosinophil, ILC3s, etc., they have inducible expression of class 2, but non-professional cells have class 1. They can use class 1 and lack co-stimulatory. Atypical APCs, antigen presenting function is limited to specific immune environments only, especially in type 2 immune settings. Then they lack compelling evidence that they can activate naive. So it's not sure whether they can completely activate, they are enough to activate the CD4 naive cells uh, in an antigen specific manner. So this is about the professional antigen presenting cells, non-professional atypical antigen presenting cells. Next is alternative pathways amplification loop. So we just saw that C3 convertase amplification loop occurs. So why this is important is we get rapid and robust amplification leading to enhanced pathogen clearance. How it works is the alternative pathway operates through a positive feedback mechanism where C3b generated from any complement pathway. It is not necessary that the C3b should be from the same pathway. Any C3b works the same. So C3b generated from any complement pathway, classical lectin or alternative, can bind to factor B. They are all same C3. So factor D then cleaves factor B into BB. This is your alternative pathway. Then C3 convertase continuously cleaves C3 into more C3b. So your spontaneous C3b, the takeover occurs, leading to self-reinforcing loop, because C3 is the uh, common point in all the three pathways. This amplification loop helps in reinforcing the activation of the pathways. So the amplification loop, enhanced opsonization occurs because more C3b deposition on pathogen promotes the phagocytosis. Then accelerated complement activation occurs because the loop amplifies the response, ensuring fast and strong immune defense. Then we also have increased membrane attack complex, that is the MAC formation, because more C3b leads to more C5 cleavage because it forms the C5 convertase complex, and this leads to MAC formation where C5b along with 6, 7 and 9 forms the MAC complex, pore forming complexes, which leads to osmotic lysis of the pathogenic cells. Then antibody independent protection. So the alternative pathway can also function without the antibodies, providing innate immune defense against the pathogens. So we have here the alternative complement pathway, activator surfaces, your hydrolysis of your C3, factor B, factor D together. Then we have C3 convertase complex. So now we, once this is there, we enter into a positive feedback loop leading to amplification. So this is your amplification loop. So once this is there, your C3b will now do its different functions with C3b, C3a, C3b, with convertase complex, etc. So finally, several immune responses like phagocytosis, chemotaxis, inflammation, cytokine release, um, immune defense, phagocyte recruitment of phagocytotic cells, and pathogen lysis, pore formation, MAC formation, etc. occurs. And the classical complement pathway, so is an antibody dependent pathway because it is stimulated by the antigen antibody complex. So this pathway that plays a crucial role in the immune defense by opsonizing pathogens, promoting inflammation and forming membrane attack complex. So this is again how step by step your classical pathway is activated. So initiation is by the C1 complex activation. The pathway is triggered by antigen antibody binding, IgM, IgG, and C1 complex contains C1q, C1r, and C1s. So C1 complex binds to the Fc region of the antigen antibody complex. Then C1q activation occurs, then C1r activation occurs, and C1s activation occurs. Then formation of C3 convertase enzyme complex. So C1s then cleaves C4. C1s cleaves C4, forming C4a and C4b. So we have C4a and C4b fragments. C4a is again an anaphylotoxin, and C4b then binds downstream to the pathogen surfaces. Then C4b also binds to C2. This then is cleaved by C1s, forming C2a and C2b fragments. So C2a and C2b, uh, C4a and 2a complex, C4 sorry C4b and 2a complex is the C3 convertase complex, which then cleaves C3 into C3a and C3b. So formation of C3-5 convertase now occurs by C4b 2a binding to C3b. So C3b binds to C3 convertase, forming C5 convertase. So C5 convertase then cleaves 5 into 5A and 5B. Again, 5A is an anaphylotoxin or an inflammatory mediator, and then C5B then stimulates the MAC complex formation. So it recruits C6, C7, C8 and multimers of C9, forming a pore complex in the pathogen cell. So these components form the C5b MAC complex and create spores in the pathogen membrane, leading to cell lysis. So the cell swells and then bursts because of osmotic imbalance. You can see here the MAC complex. It's a pore forming complex. It contains all these complement components along with the multimer of C9. So together it forms a cylindrical membrane attack complex. So the key functions of the complement pathway are opsonization, inflammation, cell lysis. C3b coats the pathogens, enhancing the phagocytosis. C3a and C5a acts as anaphylotoxins, recruiting the immune cells. Cell lysis occurs because of the MAC complex, leading to pathogen destruction. So first is initiation, then C3 convertase formation, C5 convertase formation and MAC formation. These four are the four most important steps that take place in the classical pathway. So that is it. So this topic is the alternative classical and lectin pathway, this week's syllabus mainly. So how its regulation occurs, positive feedback loop occurs, how it helps in enhancing the inflammation, how it helps in enhancing the cascade, how C3 convertases are different in case of classical alternative lectin pathways, and how MAC complex formation occurs. And finally, clearance of the pathogenic cells via the cell lysis, phagocytosis, etc. This is what consisted in this week. So this is a gist of the three pathways which are major and most important for complement system. So you can see spontaneous hydrolysis here and alternative pathway, then factor D, factor B, then C3 convertase, then C5 convertase. Also opsonization occurs, recruitment of phagocytic cells by C3b binding on the pathogens. Then we have the amplification feedback loop, which helps in continuous alternative pathway activation. Then we have classical, which has the C1, C4, C2, and we have lectin, which is mannose binding lectin, binds to the mannose residues or carbohydrate residues. Then we have picolins and MASPs, which then again form the C3 convertase, and both C3 then help in formation of C5 convertase, and C5b helps in membrane attack complex formation, where C5b binds to C6, C7, C8 and multimer of C9, forming a pore complex in the pathogenic cell. Then C5a, C3a, other than this, are the anaphylotoxins or inflammatory mediators. They trigger inflammation. So this is your basic complement system with three pathways: alternative pathway, classical pathway and lectin pathway. So that is it for this week. Hope this was useful. It's a little repetitive because many of the questions were relative and it was connected, so I had to repeat few of the things, but it helps in remembering all the three pathways efficiently. Okay, I think one left. So if there are any doubts, since many people are not attending the sessions, you can always check out the videos and mail me to this mail ID if you have any doubts or queries. And that's it for this session. Thank you.